Good morning. In this presentation, we'll be reviewing the methods of scientists and how they relate to scientific investigations. All right, let's go ahead and dive in. All right. So first, I'd like to look at this flowchart. Take a moment, look at this flowchart, and this is a standard structure that you could use to do a scientific investigation. And as you can see, you'll first make an observation, and you're going to ask a question about that observation. And then regarding that question, you're going to form a hypothesis that might answer the question. And then with that, you're going to run through this process. And if you find eventually that your hypothesis is incorrect, you might try again. And you'll loop around until your hypothesis is correct and report the results. And also sometimes you may actually loop back from a correct hypothesis, but it just depends on the situation. So scientists use similar methods to gather data and communicate information. Scientific methods are a series of problem-solving procedures that help scientists conduct experiments. There's many other methods that might not be scientific, but we are talking about those methods that happen to be scientific. A hypothesis, specifically a scientific hypothesis, is a testable explanation of a situation that can be supported or disproved by careful procedures. You may sometime in your lives have gone to science fairs. They're pretty popular in junior high and in elementary school. We don't do them as much in high school for some reason. I'm not really sure why. But you may have seen different experiments where you'll compare, for example, the growth of two plants, and you'll have different situations, different variables. And we'll talk about the types of variables that may affect the growth of plants or may affect any other experiment that you might have. It's easy to relate to plants. So this particular slide, I'm going to relate to plants, but just recognize this doesn't only have to do with plants. Okay, so here's an experiment that appears to be corn, but this probably applies to any plant and any culture for that matter. You could actually do cell cultures and follow this procedure. So while I'm going through this, I want you to see if you can identify from your memory what the independent variable is, what the dependent variable is, and what the control is. We haven't quite gone into those yet. Okay, and maybe I'll just jump forward for just a moment, just to remind you, the independent variable is, in an experiment, is the factor that is changed by the experimenter. So this is you. Your actions are the independent variable. And typically you won't want to have very many actions that can behave as an independent variable. So you'll usually want to limit that to one thing. The dependent variable is the factor that is affected by the changes in the independent variable. So in other words, the things that you do as the independent variable will cause a change in the dependent variable, but it's not direct. Okay, and then the control is a thing that's the same along the whole way. Okay, let's go back to this slide and look at these different situations and see if you can identify the independent, the dependent, and the control. All right. So in this first picture, we have just distilled water, or just deionized water, which is water absent any type of minerals or any type of nutrient, anything like that. It's just pure water molecules, essentially. And in the second picture, we're adding nitrogen, so some type of fertilizer that only has nitrogen. This is only fertilized with iron, so no nitrogen. This only has magnesium. So these all have distilled water. Okay, this one is distilled water plus nitrogen. This one is distilled water plus iron. This is distilled water plus magnesium, distilled water plus phosphorus. And when it says full nutrient, they're saying distilled water plus all this stuff. And you can compare the growth in all these. Okay, so you should have been able to recognize that the control was distilled water. They all had water. Without water, we wouldn't have had any growth, right? Plants and most life, if not all life, depends on liquid water. Some life is able to derive water from their food, but they still need liquid water. And I've heard that there's some plants that can actually pull moisture out of the air, so that's possible. I don't know anything about that, but I've heard that that's possible. Okay. Usually I would guess what would happen is you might have water vapor condense on leaves of a plant, which is turning it into liquid water. So I don't know. If you guys know anything about that, go ahead and leave a comment and teach me. So in this case, our independent variable is the thing that we're adding. In this case, we're adding nitrogen, iron, magnesium, phosphorus, full nutrient. Those are all our independent variable. And because of our independent variable, we can observe different amounts of growth in our dependent variable. Our growth is dependent upon our independent variable. Okay, so we change the nutrients, our dependent variable, our growth or nature is responding to whatever we gave it. In this case, we're watching our dependent variable on this particular one win with the most growth. These other ones are all yellowish leaves because they don't have any nitrogen to make chlorophyll, probably. That's just speculation, I don't know. So there you go. Back to this, our definitions of our independent dependent control. I'm not going to stay here very long, but you can see that you can independently do other things with plants. Give one plant water, give one plant juice, give one plant Coke, see what happens. Okay, here's another controlled experiment. In this particular experiment, the only different factor is water. So we're using 10 beans per pot. We're using the exact same pots, the exact same substrate, the exact same dirt. And then in one of these, we're going to add water, and one of theirs we're not. We probably should have said how much water we're adding to the one we're adding water, but we're going to ignore that for now. And then over here, we can actually see a germination rate. So I imagine that most seed companies are probably going to do some type of experiment similar to this, so that when they're selling their product, they can report what their germination rate is. So for example, if this was their results, they would probably report a 90% germination rate. And that's useful. If you happen to be farming and you have a large plot of land, it would be useful to know the germination rate so you know how many seeds to use. In this case, you might want to use 10% extra on your seeds to compensate for the germination rate. Okay, in this case, we can have some controlled experiments where we are analyzing different fertilizers. And our control isn't using fertilizer. This is a very useful experiment. And you would obviously want to make sure you have the same dirt, the same soil being used in all these experiments, because there are some nutrients just present in the soil, or you could do this hydroponically with just water. So this is an interesting experiment. In this first, in the control, we are absent fertilizer. In the first one, we have this fertilizer brand X. I love how they didn't use a brand name on this one, because obviously they don't want to make anyone look bad. But if you were to discover a fertilizer that inhibited growth, you probably wouldn't want to purchase it. And if you spent a lot of money on it, you probably wouldn't be very happy. So if you were a farmer, it might be useful to do some type of a controlled experiment with some of your different options for fertilizers, so you can see how they may or may not affect your fields. And for example, if these two theoretically had the same price, I probably would lean toward the one that had more growth. And if the growth wasn't that much of a difference, I might question whether or not I needed fertilizer. In my yard, I don't really try to use fertilizers. I mean, every once in a while I might add some if I have any yellowing of leaves, but if I don't observe any malnutrition in my plants, I try not to add fertilizer because I'm trying to create a symbiotic relationship with different plants, but that's beyond this. Another example is this graph right here. This graph is taking the results of our experiments, and by putting them into a scatter plot, we can derive more information. We can make inferences, and we can do what's called interpolating or extrapolating based on our results, and I'll explain those in just a moment. So I want you to look at this graph before I go any further, and I want you to see if you can figure out which one of these is the dependent variable and which one of these is the independent variable. So I want you to read the titles for the two axes and determine if the x-axis, which is along the bottom, if that's the independent or the dependent variable, and then if the y-axis on the left side is the independent or dependent variable. So think about that for a minute. Pause this if you need to. See if you are able to do that, because I'll probably have a test where you need to look at a graph and tell me which one is the independent variable and which one's the dependent variable. Okay, so go ahead and pause. All right, I'm gonna assume you've answer that question. So looking at this, the amount of water, daily amount of water. This is what we've done. We've added this amount of water. The thing that we've done is adding a certain amount of water. In plant 1, we added 0 milliliters. In plant 2, we added 2. Plant 3, 5, then 7, then 10, etc. That's what we did. That's our independent variable. Our dependent variable is what the plant did in response to what we did. So we added the water, the plant did the growth. We can't directly tell the plant to grow. We can't force the plant to grow. We can add water and then we can wait and see what happens. And that's what we've done here. The one where we added 0 water, surprise, surprise, no growth. And then where we added 2 milliliters, we got 2 inches of growth. 5 milliliters, we got 3 inches of growth. We had a huge spike here, 7 milliliters of water, we had 6 inches of growth. And then we see something interesting. As we continue adding water, the growth actually decreased. We still had growth, but the growth decreased. This goes along with that whole, if a little bit is good, more must be better. It's obviously not true. Too much water is causing a decrease in growth. And you can speculate as to why. You would need further experimentation to actually prove this scientifically. But I would probably hypothesize that by adding too much water, we are putting the roots into an environment that has low oxygen, a hypoxic environment. And a lot of times when you deprive parts of plants of oxygen, it inhibits growth. So we probably had a decrease in root growth. And by decreasing the growth of the roots, it's going to affect the entire plant. And I would actually further speculate that you could extrapolate, which means that you go past 10 milliliters. Even though we haven't experimented past 10 milliliters, you could probably eyeball this and extrapolate, which means you're making a scientific assessment of where this plot is probably headed as you add more water. And if we assume a linear plot, which it might not necessarily be, you could probably eyeball it's probably somewhere around 15 milliliters maybe. If you were to theoretically add 15 milliliters, you may or may not drop down to 0 growth. And that's called extrapolation because we don't have any results past that point. If we wanted true results, we would continue experimenting past that point and figure out where we get to the point where there's 0 growth. Okay, and there might be diminishing returns, so it could be anywhere out to 20, 30 milliliters, who knows? That's called extrapolation. And if you want to interpolate means you try to guess, you try to hypothesize the data in between 2 points. So for example, if you wanted to look between plant 4 and plant 5, we didn't do an experiment at, where is that, like 8.5 milliliters. We didn't do an experiment there, but we could interpolate that the growth would be about 5 inches at 8.5 milliliters, okay? So you've just learned about extrapolating, where you're outside of the bounds of your experimentation, or interpolating, which is in between, all right? But like I said, that's just a scientific hypothesis because you're basing that off of other results. And if you have enough data points, you could even say that that's a theory. So it depends on how you look at that. But that's just interpolation is in between, extrapolation is outside. Okay, so once again, this is our pattern. This is a flowchart. A lot of times if you're a programmer, you could actually use flowcharts to have a high-level interpretation of what your plan is. And then by using that, you could guide yourself into what is important that you add at the low level when it comes to programming. Okay, and you have already started an experiment like this, a lab in this class, or at home, where you're supposed to write down your directions on how to get from point A to point B, kind of like a recipe. And you'll notice that if someone's following your recipe, if you had any ambiguity or any lack of clarity, they might not end up where you planned on them ending up because you didn't use words that were unambiguous. Your words had a potential of more than one plausible direction, and so they may have not ended up where you planned. So that's the reason I wanted you to do that, so you could realize how important it is to be as clear as possible and unambiguous as possible. All right, so that's what I wanted to cover in this, and thanks for watching.